Okay, all
Go
right.
ahead.
So hopefully everyone can hear me. It's my pleasure to introduce our main speaker today, Dr. Suneil Koliwad, who is both a clinician and a researcher at University of California at San Francisco. He's the chief of endocrinology and metabolism and the Gerald Grodsky PhD Distinguished Professor in Diabetes Research. He's also the Mount Zion Kenneth Werber Distinguished Professor of Endocrinology and Metabolism. He has a long history of involvement with the American Diabetes Association, both locally and nationally. He's nationally recognized, was a member of the writing group of the ADA Diabetes Standards of Care back in 2015 through 2018. The diabetes standards of care inform the care that's delivered at every hospital throughout the nation. And he's also been a member of our peer review research panel that selects the researchers for ADA funding. He is the director of the Koliwad Lab at UCSF, which is at the studying at the intersection of nutrition, immunity, metabolic health, diabetes, and obesity. Dr. Koliwad is going to share some exciting information today that impacts both diabetes type 1 and diabetes type 2. And without further ado, Dr. Koliwad, I'm going to turn it over to you.
Thanks a lot, Tom. I appreciate it. Can everybody hear me okay? Wonderful.
Yes.
Great. Well, thanks to Danny and to Alan and Tom for this really kind invitation, and to all of you for being here. I've done this before a few times, and I've gotten kind of used to, as so many of us have, to doing these sort of things virtually now. But I do have to say that I really miss the opportunity to be in physical space with folks interested in learning more about diabetes, helping us stop diabetes, and being a part of a community, which I feel like I'm also a part of, not just because I work on diabetes and have devoted my entire career to lessening its burden on our society, but also because, like you all, I'm a resident in the Bay Area, raising a family here, and really love the fact that the Tour de Cure is this wonderful way of, in my opinion, integrating what we all love about where we live with this important issue that is top of mind, not only here locally, but across the country and across the world. And it's for that reason that I am a really avid participant in activities run by the ADA. And it not only helps fund my laboratory and the initiatives in my diabetes center, but it also provides me with a way to engage my passion for diabetes at the community level. And hopefully over the next few minutes together, I can share some of that with you, interspersed with a little bit about the science as well. So why don't we just kick off? I've got a few topics to go over, and trying to keep things fresh, and let you know that despite the pandemic, and maybe even in some ways because of the pandemic, research in diabetes and its underlying drivers like obesity, as well as its complications, has continued aggressively and with a lot of energy over the last year plus. And I'll try to hit a few highlights about what we're learning. So I'm going to start out with a little bit of a time spent on diet. I think that you all clearly have gotten the message by now, and probably many of you are putting this message into practice, that our patterns of dietary intake have a strong influence on our metabolic health. And so we think about a heart healthy diet. We think about diets to limit the likelihood of developing diseases like type 2 diabetes in particular. And just as an aside, to make sure that everybody's on the same page, type 2 diabetes is an elevation in blood sugar which is a common feature of diabetes mellitus, no matter what type you talk about. But this elevation in blood sugar arises not because our body is incapable of making the key hormone that controls blood sugar, which is insulin, but rather because in genetically susceptible individuals, especially in the context of poor dietary history over time, as well as potentially inactivity and associated obesity, the body's ability to respond to the signals that are dictated by the hormone insulin drops off. And this lack of responsiveness we call insulin resistance. And insulin resistance is a precursor of type 2 diabetes. And type 2 diabetes, as many of you are aware, depending on what region of the country you're in, can affect, from anywhere from 15 to 20%, all the way to 35+% of the population. And in some places in the country, nearly half of individuals, adults, have what we call prediabetes, which is this insulin resistant process that leads to an increased risk for type 2 diabetes. And type 2 diabetes accounts for 90%-ish of all of the diabetes. And so preventing type 2 diabetes and stopping the progression of type 2 diabetes once it onsets is a huge public health concern. And everybody living in the United States is likely to know somebody who has or is at risk for developing type 2 diabetes. It is sparing nobody. And so with that in mind, diet is really important. So what have we learned about diet in the last few years? And I think the most relevant snippet that I can add, in addition to other general tenets of healthy diet, is the real central importance of limitation of simple and processed carbohydrates. And no more has this become evident, the value of this limitation of carbohydrates, than in the recent studies looking at intermittent fasting and looking at ketogenic diet. And so what have we learned about these two types of diets? So ketosis is a process where the body generates a fuel itself in the liver called ketone bodies. And ketone bodies are an essential way to keep us alive, alert, and functioning when we have not eaten in a very long time. Usually, carbohydrates, glucose, is our body's preferred form of fuel. All the cells in our body are capable of using glucose as a fuel. And insulin, the hormone, which the pancreas makes and secretes into the bloodstream after we eat, is a signal that we have eaten, that there is glucose coming into our system. And like a key, insulin acts to unlock doors in each of our cells that allow the glucose in our bloodstream to go into the cells, where it can be used as energy to fuel that cell's functions. In particular, the brain cannot use fat as a fuel, whereas other tissues in the body, muscles, for example, can use fat as a fuel. And the brain's reliance on glucose makes it very vulnerable to succumbing to a lack of glucose that might happen when we have not eaten in quite some time and we've been very active for all that time. And so you see this sometimes in marathon runners who don't appropriately carbo load before their marathon. They'll be, or bicyclists. Marathon runners can hit the so-called wall late in the race, and cyclists, and I hope this happens to nobody who's participating in the Tour de Cure, can engage in something that cyclists call bonking, which is what happens again when the brain is devoid of enough glucose to fuel its function. We can't go for even a few minutes without having sufficient fuel supply to the brain. So if you do carbo load enough, then you're able to allow your liver enough time to generate something we call ketone bodies. And ketone bodies are a fuel that the brain can use. And what we've learned is that when you eat a diet that is devoid in simple carbohydrates, devoid largely of carbohydrates in general, in particular simple carbohydrates that are so common in sweet foods and processed foods, you engage a little bit of this ketone body production by the liver, and intermittent fasting does something very similar. And so that's why we call that carbohydrate limitation diet a ketogenic diet, is because it facilitates the liver to make ketone bodies. And intermittent fasting, because we're not eating anything at all for those prolonged periods of time, does the same thing. And what we've learned in the last couple of years is that ketone bodies have an immense health benefiting effect on tissues throughout the body, including our muscles, the heart, the brain, and that by generating a little bit of this ketosis, we can not only limit the development of diabetes and cardiovascular disease, but in animal models, at least, can prolong life itself and improve lifespan. So the name of the game now is to try to figure out all kinds of different ways to allow our bodies to generate just a little bit more ketone bodies and ramp that up. And I'll conclude with the fact that there are two, well, at least one in particular, newer class of anti-diabetes medications for type 2 diabetes called SGLT2 inhibitors that include medications, the brand names of which are Jardiance and Invokana and Farxiga, for example, that actually produce a little bit of this ketosis by taking the medicine. And we're trying to learn how that works, and that's an active area of research. But that little bit of ketosis that this medication produces is associated with benefits to the kidney and benefits to the heart and its function in people with diabetes. And so we're actually trying to now pivot to thinking about SGLT2 inhibitors as kidney protective drugs and heart enhancing drugs, even in people who don't have diabetes. And so this ecosystem of learning how diets work at the molecular level, figuring out ways to replicate what those diets do, developing medications that can try to do some of that, and then studying those medications, that has been an ongoing cycle of scientific pursuit for decades now. But in particular, with respect to ketone biology, that has been one of the things that has hit the fore in the last couple of years. So let me pivot now from diet to another area that has really become a very important one, and that is what we call precision medicine. In this case, precision diabetes management. So one of the things, of course, that precision medicine sort of aspires to is to reach a point where we decide in a customized, personalized manner how we are going to treat each of our patients based on specific individual factors that define that person. Right now, we just say, well, you have type 2 diabetes, and so here are the medicines for type 2 diabetes, and we try them one after the next, and we try to find ones that work. But what if we could find, through specific blood testing, perhaps non-invasive imaging, perhaps genetic analysis, and perhaps even other variables that are taking into account questionnaire-based responses, demographic features, ethnicity, and the like. What if we could come up with ways to accurately predict what medicine is likely to work for you right off the bat, so that our likelihood of trying and succeeding with our first choice medication versus trying and failing and having to try again and again and again would go way, way up, so people could get under good control as quickly as possible? Well, that seems aspirational, but precision medicine holds that there are technologies in place right now that can get us to that point in a very palpable manner. And it intersects with diet, because after all, one of the things that we feel like defines us as individuals is that we have the freedom to choose what we eat, and everyone eats something different. We don't all eat exactly the same thing. And we're making choices every day about diet. But what if we could have specific blood testing, specific analysis of our genetics and other factors that could help each individual person determine what might be the best set of choices dietarily for them, with the idea that what is perfect for you, an optimized pattern of eating for you, might not be what is the optimized pattern of eating for your spouse or for your friend down the block or for your colleague at work. And I think that's the future. That's where we want to go in the next 5 years, is to get the ability to use data to predict what each individual might benefit from the most with respect to patterns of sleep, patterns of exercise, patterns of eating, and medication choices. And so that is precision medicine in a nutshell, and it's being aided by a revolution in what we call Omix, which is large-scale, whole genome, whole proteome, meaning all the proteins that the human body makes, whole secretome, all the secreted factors that are circulating in the blood at any given time, and the ability to analyze all of these protein factors and secreted factors and genetic and genomic factors simultaneously, using very advanced computing technologies to take in all that data, deconvolute it, and provide meaningful information about how the human being functions. And then point all that omics technology towards diseases like diabetes. And you see now paper after paper after paper trying to optimize the use of omics in multiple platforms to get deep understanding about specific, precise, individualized responses to various interventions in the space of diabetes. And I think that you're going to see over the next 5 years, this is really becomes the mainstay in research. And over the next 10 years, all of our clinical lives are going to be influenced one way or the other by the ability to do more precise guidance for how we should be engaging in our preventative and management strategies for diabetes. So all of that really is linked to and most advanced in the area of type 2 diabetes, like I mentioned. But there is another type of diabetes that is worth talking about in detail, and especially because this is a very important year. I don't know if we've discussed this with this audience yet, and I don't know if you have had this on the top of your minds, but 2021 into 2022, right now is the 100th anniversary of the discovery of insulin. And in my mind, the discovery of insulin is probably one of the real miracles of medicine, not just modern medicine, but medicine in general. Because prior to the discovery of insulin, patients with type 2 diabetes who got worse just kept getting worse over decades, and there was nothing that we could really do to ultimately control their blood sugars because we didn't have insulin as a backstop to treat them with. And insulin is almost universally effective in people with type 2 diabetes as a way to control blood sugar when nothing else can. But for type 1 diabetes, which is a disease not due to insulin resistance, but due to the fundamental inability to make insulin, insulin as a medication is essential for life. And prior to the discovery of insulin, type 1 diabetes was uniformly a fatal disease, and people would die within a matter of weeks to months after their immune system began its autoimmune or self-pointed attack on that individual's own beta cells, i.e. the cells that make insulin within the pancreas of that person. So our immune system is exquisitely designed to recognize what is self and what is non-self, and attack that is non-self, bacterial invaders, viruses, parasites, and other pathogens, but spare our own cells in the process. In individuals with type 1 diabetes, as an example of a so-called autoimmune disease, that self-recognition element goes awry in a specific way, and the immune system becomes confused and misrecognizes, specifically in this case, the cells that make insulin, unfortunately, as foreign, and attacks them and ultimately destroys the insulin producing capacity of the pancreas. So where are we in the last couple of years with respect to circumventing this in a fundamental way? We've been able to give patients with type 1 diabetes insulin, better and better and better types of insulin, and using technologies to create continuous delivery modalities to allow patients to have exquisite control over how they give themselves their insulin with a minimal amount of physical work necessary versus decades past. the ability to continuously monitor glucose, and in closed loop systems, respond to that monitored glucose by providing insulin as much as needed and just when it's needed, in a manner that allows the individual in question to be passive and going about their business more so than constantly being focused on their blood sugar checking. But beyond this so-called technological fix for diabetes, where are we with fundamental biological fixes for diabetes, the so-called cure, as in what we're all writing for very soon in the Tour de Cure? Well, gratifyingly, this has been an important year in that regard. 100 years after the discovery of insulin, we had the very first new therapy for type 1 diabetes go before the FDA and actually get approval by the initial advisory panel from the FDA. Now, this medication in the end may not be approved by the full FDA, but it is a watershed moment, and I think we should all appreciate that this is not going to be the only thing that comes, and there's going to be a silence for another 4 or 5 years before the next thing comes. This medication, which is an antibody against a type of T cell expressing a molecule called CD3, adjusts the balance of the immune system and tunes it in a way that this autoimmune attack is mitigated. And that monoclonal antibody was shown to be effective in people diagnosed early in the course of type 1 diabetes, with staving off the complete loss of insulin production capacity, so that individuals who were in that boat could go several months, if not at least a few years, before they ever needed to start taking insulin. And that ability to delay substantially the onset of diabetes per se is revolutionary, because it tells us that we now have the right track in terms of how to attack the immune system in a way that can modulate the onset of this disease. And with that, we can go after 10 more targets today to try to replicate and build on that successful demonstration in people, not just in animal models. And so this was a really important year for the immunology of type 1 diabetes in that regard. And I think with respect to the other aspect of this is to how to regenerate the pancreas, right? And so there you employ two aspects of the cutting edge. One is stem cell biology. How do we identify cells that we can get to turn into insulin producing cells, and then populate them back in the individual who lacks them, to give them back that function? And then how do we protect those new insulin producing cells from being destroyed by the immune system, which is going to misrecognize them just like it did the ones that were there originally in that same individual? So that leads to a combined immunologic and stem cell oriented encapsulation strategy, which is a form of the so-called artificial pancreas that utilizes one's own stem cells that can be now turned into stem cells from a skin cell or a blood cell, what are called induced pluripotent stem cells, and then an algorithm and a treatment strategy to coax those stem cells to turn specifically into insulin producing beta cells. And then once they are that, and we have vetted that they are bona fide in that capacity, coming up with a nanotechnology oriented approach to encapsulate those beta-like cells, put them into the body, let them make insulin in response to glucose like the normal pancreas would, while they're protected from assault by the immune system. And we can come up with ways to modulate the intensity of the autoimmune response, as well as creating an effective encapsulation strategy so that those immune cells can't get in and get access to these insulin producing cells, but the insulin that those cells make can get out into the bloodstream and do what it's supposed to do. we are getting closer and closer, both on the stem cell front. There have been several papers in the last 2 years published, including during the pandemic, indicating that we keep ticking off more boxes where these cells pass yet an additional test of similarity to mimic what a real beta cell can do. And at some point, there are going to be no more boxes left to check and no more hurdles left to overcome. And we'll be able to say that these are bona fide beta cells. You cannot tell them apart from the ones that an individual carries in their pancreas from the get-go. And that day is basically on the verge of being here already. And then the nanotechnology approaches, this gets into the technology component of all this. In addition to the closed loop system of artificial pancreas, this type of encapsulation-oriented artificial pancreas is really great because it's a permanent fix if it were to work, and you would not need to change out anything, to refill anything, or to replace any batteries and that sort of thing. And so the good thing about the technological artificial pancreas and this biotech artificial pancreas approach with stem cells and encapsulation is that it involves collaboration between NIH and American Diabetes Association funded research initiatives and labs in academic centers across the country and across the world with private sector industry. And it's that private sector industry component, very relevant because we're here in Northern California, which includes Silicon Valley, which is such a hub for this kind of technology. The competition inherent in industry and its approach to this is absolutely essential to the pace of the progress that we've made. And because we have multiple companies competing in this space, the advances are coming faster and faster and faster because people want to be first. And that is great. That is just great. So just to hit the highlights there in terms of technology, I think that we are developing better and better approaches and more simplified approaches at glucose monitoring that are smaller, more continuous, more easy to access, more easy to share with family members and share with your doctor. And that is fabulous. Better technologies to then link those glucose values with insulin delivery systems that are increasingly smaller, more nuanced and responsive and less cumbersome to the individual. And then approaches to try to rebuild the pancreatic glucose management system in the body biologically by controlling the immune system and using stem cells with encapsulation approaches to accomplish a reconstitution of a pancreatic insulin producing function and therefore then cure what was lost before for the person with type 1 diabetes. So before I conclude this part in a minute or two, just want to say two more things. One is that I think it's incumbent on all of us to realize that that with all of what I just talked about, we also have to constantly have an eye on equity and equitable distribution of the best dietary practices, precision approaches to diabetes, immunological advancements, and novel technologies so that everybody gets to benefit from them. And there are no subsets of our population who are ultimately left out of these advancements and don't get the benefit of all these tools to improve diabetes. My goal and the goal of my colleagues throughout diabetes research and clinical care is to make sure that we spread this wealth of understanding for everybody. And I think that with that in mind, the American Diabetes Association is really notable because it has a huge component of its mission to include advocacy for people without a voice, to make sure that that underserved populations and vulnerable populations get educational resources and the benefit of all of the types of research advancements that I just discussed and more. That we use creative techniques to work through, for example, African-American churches as a means to disseminate important information to communities of color. And in the end, we fund initiatives, grassroots initiatives, as well as national strategies to reduce what we call disparities in diabetes care and disparities in diabetes management. And there's just no way that we're going to get where we need to get, despite all of the advancements in science that I mentioned, unless we create effective strategies to reduce disparities, because our healthcare system and our country in many ways is exemplified by the disparities that exist every bit as much as it's exemplified by the amazing achievements that we have as a society. And so for diabetes, that's just a non-starter. We cannot allow that to be what rules the day. And the American Diabetes Association, and why I'm really so proud to be involved with the ADA, has always felt like, you know, it needs to be a leader in this area. And I just want to make sure that you know, it's clear that element of equity and inclusion is always front and center in how we advance things in diabetes. And then finally, just a quick point to say that, and this is going to lead to hopefully a little fun interactive piece before I sign off here. I want you to know that by being involved in the Tour de Cure, you're accomplishing a lot more than you might realize you're accomplishing. Firstly, you're helping to fund a real ecosystem. I talked about the disparities piece, advocacy, education, empowerment, all these other aspects of what the ADA does beyond just funding laboratory research and clinical studies and trials, which it also does. But the other aspect of this that I think you may not realize is that it really is the case that every dollar counts. I'll give you an example of this. I have there was a postdoctoral scholar who, after completing her endocrinology fellowship and wanted to do a career in diabetes, or was considering one, joined my lab. She is a Hispanic Latinx woman in science, and you know, we don't retain enough individuals from vulnerable populations like Hispanic Latinx individuals fall into, and we don't retain enough women in science in general. And so when you see somebody who you think has what it takes to be a real contributor in diabetes research, who falls into those categories, you would love to be able to retain that person in academia if you can, and influence them to devote their career to diabetes like I did. And she got an American Diabetes Association postdoctoral minority fellowship. She got this award in 2016, and it funded her for 3 years. After that, she was able to get a Robert Wood Johnson Minority Faculty Advancement Award, and most recently has been able to pivot that into getting an assistant professor faculty job position at UCSF to do her own diabetes research, which is interestingly focused on diabetes and risk factors across diverse ethnic populations, which is absolutely perfect given her background. And I think that you know, you might say, well, small potatoes contributions don't really do the trick here. They do, because this was not a big amount of money to fund one postdoc, but that one postdoc decided to stay in diabetes research for a career because they got that award. And now the diabetes community will get the benefit of 25 to 30 more years or more of research from that junior scientist that might never have happened had it not been for that small amount of funding. And so I think it's really important. My lab has benefited from the ADA directly in this regard. And then with respect to the ecosystem, it's also notable that your dollars with the ADA, they don't just go to ADA nationally and fund science in dilute ways that you may not appreciate palpably. But the scientists who you help fund are local. I live here in the Bay Area. I'm one of your neighbors. I raise my kids here, and I'm part of this community. And there are a few scores of scientists who have benefited directly, who I know personally, in diabetes and related diseases that are linked to diabetes and its complications from ADA funding, who live right here in the Bay Area at UCSF, at Stanford, at Berkeley, at the Buck Institute, at UC Santa Cruz, at UC Davis. And we have a network of scientists here locally who use ADA funds in a variety of ways to do everything from the most basic cellular and molecular research all the way to community-based interventions in human populations and in specific communities. And so when you do what you're about to do here and ride in a few weeks, that is actually funding research that's going on right in your own backyard to benefit diabetes, as well as it's going on all around the country. So I just wanted to leave you with that message. And with that, see if I'm going to unhook my computer. Hopefully everything stays good. It did yesterday. So I think you can all see me, right? So this is just a quick tour, just letting you know, even though it's the pandemic, I'm going to put my mask on because I'm going into the lab and leaving my office. And I'm really happy that my lab is connected to my office. And so I'll take you out here and just let you know that even though a lot of stuff is proceeding virtually, lab work proceeds in person. It always has, and it always will. And so now you're in my lab, and my lab is focused on obesity and diabetes, including in people as well as in animal models. And so there's one of our postdocs, Reed Bolus.
Hi there.
Hey there. And so Reed came to our lab from Vanderbilt University, which is another fabulous diabetes oriented research center that has led the way in many aspects of diabetes. And we're thrilled. Reed is working on diabetes and its effect on the liver and fibrosis and a process called fatty liver disease, which is very common in people with diabetes. And then Diana Alba here. Hey, Diana, is assistant professor at UCSF and a diabetes researcher who's also based at the San Francisco General Hospital and runs the obesity and diabetes clinic associated with that hospital, and is the individual I told you about who got the American Diabetes Association postdoctoral fellowship early in her career. Other folks are in adjacent rooms doing cell culture and experiments, and we're having lab meeting with some of those folks later today. And I am thrilled to tell you that, I'm just as energetic about focusing on diabetes now as I have been at any point during my career. And even though the pandemic has posed a lot of frustrations for us in science and clinical care, as it has for all of you in life in general, one thing that hasn't really wavered is our commitment to keep cracking at diabetes and keep making progress. And so hopefully this was really really valuable for you. I know it has been for me, because I've done this in person prior to the Tour de Cure, and I'm glad to have had the chance to at least do it virtually this time around. And hopefully I can see some of you closer to race day. And I'm thrilled that you're all so interested in being involved, and I hope your ride goes super well. In the last couple of minutes here, let me just go through some of the questions that came in through the chat while I was speaking. So there's a question here about artificial insulin. So I think I covered some of this, but the question is, for those who already have diabetes or a loss of insulin production due to pancreatic cancer or removal of the pancreas, outside of insulin pumps and CGM systems, how far away are we from an artificial insulin? I think what the person probably means is a re-engineered capacity for the body to make insulin again. And I think I went over that. That's really a stem cell oriented approach, which requires arresting the immune system in a specific way and encapsulating those and protecting those stem cells so that they can function for years and years and years without being worn away by the immune system's attack on them. But just in case, there was a question. The insulins that we use, whether they're in a pump or whether you inject those insulins manually, they are artificial insulins now. For those of you who are older with diabetes or know somebody who's older with diabetes or who may have passed away, long ago, initially we used animal-derived insulin that was actually obtained by, extracting it from the pancreas of animals like cows, and that bovine insulin held the day for decades prior to the advent of molecular biology and cloning, which allowed us to clone the gene for insulin, which was interestingly enough done at UC San Francisco, my institution. And after cloning insulin, we started to be able to make so-called recombinant human insulin and facilitating bacteria to make human insulin, so that the insulin that we inject is indistinguishable from our own, even though it was made in a bacterium. And then more recently, to make mutations in that insulin so that we make artificial forms of insulin that have enhanced properties in specific ways so that they work faster than natural human insulin or work longer than natural human insulin. And those are the mainstay types of insulin that people take now. So even without invoking some of the stuff I talked about, just the insulins themselves are amazing advancements over the last 15 to 20 years. I mean, the properties that modern insulins have, maybe people take for granted, are absolutely amazing. I mean, you can take them within a minute or two before you eat your food and it starts working. We did not have insulins that could do that 20 years ago, and now we do. So that's an aspect of quote unquote artificial insulin that's also an aside. And then so one of the question also is, are patients with type 1 diabetes automatically immunocompromised? Should they be included in getting in on the COVID booster shot type? And what about that for type 2 diabetes? So that's an interesting question. The type 2 diabetes component has been relatively easy to answer because type 2 diabetes, as I mentioned at the outset, intersects with obesity in a specific way. Not to say that all obese people will get type 2 diabetes. That's not true. And not to say that all patients with type 2 diabetes are obese. That's also not true. There's a wide diversity, but there is an overlap between obesity and type 2 diabetes. And there's an entity called metabolic syndrome, which predicts diabetes and cardiovascular disease in people with a certain form of obesity and inherited a combination of genes that predisposes them to type 2 diabetes. These people with this flavor of type 2 diabetes, metabolic syndrome, and sometimes obesity have been shown from the very beginning of the COVID pandemic to be highly at risk for severe COVID outcomes, including hospitalization, going into the ICU, and ending up on a respirator. And so recommendations for aggressively managing people with type 2 diabetes for their glucose, controlling weight, and focusing on diet for people with obesity have never been more important, because now COVID is another risk for those individuals if we don't get that control in place. And then the other thing is that we are more aggressive with dexamethasone, steroid treatment once you do get COVID, more aggressive with monoclonal antibody treatment once you do get COVID, and more aggressive with hospitalization and our therapeutic approach in the hospital to improve respiration if you have type 2 diabetes and you get COVID. So with that in mind, I believe, yes, that with respect to a third dose of vaccine, patients with type 2 diabetes will be among those groups prioritized to receive that third dose. As it is, everyone's going to end up receiving a third dose if you've gotten 2 already, whether it's Pfizer or Moderna. And so, this isn't even going to be exclusive to people with diabetes or of one form or the other, because just the time lag is so fast now. I mean, September 20th is going to be here before we know it, and everybody's going to be getting the third dose if they want it, which I think people should do. But with type 1 diabetes, it's less clear, because type 1 diabetes is a disruption of the immune system. Truly, it is, but it's not an immunocompromised disruption. In fact, it's an extra activity of the immune system. In addition to attacking things that it should attack, it's attacking things that it should not attack. So we try to damp down that immune response to allow the beta cells to exist and not be destroyed by our own immune system. However, do we know that only people with fundamental immunocompromise are at risk for COVID? We don't. It might be the case that people with autoimmunity and altered immune system function are also at risk for COVID. And with that in mind, and with new research that's coming along in the last few months suggesting that this is the case, I would advocate for people with type 1 diabetes also getting a booster, a third dose. And I'm pretty sure that there'll be, a uniform agreement that is also important as well. Finishing up here, I think we talked about the artificial pancreas. My guess is that we're going to get closer and closer over the next 5 years. I think each year in the next 5 years, there are going to be small but really noticeable advancements towards this. And that within the next 10 years, we will be on our way to the first group of people who have these long-lived artificial pancreas mechanisms. And there's going to be the two versions, the fully external technology-oriented one and the more biological, technology-driven biological implantation. And they're both going to be competing neck and neck to see who's perfect and who can do this the best. I think by 2030, that's going to be one of them. Pertinent to the next question that someone asked, and this is the next to last question, that we are going to have a long-term capacity to arrest diabetes for type 2 and type 1. And we will have a really, really tight understanding of personalized approaches to prevent diabetes that are relevant across the population. The part that I worry about, if you want to be frank, is not the science and not the technology and not the advancements of where we can get. But I worry about our ability to make these things available to everybody. That's really what I worry about, because we've seen, so frequently that we have wonderful medicines, but I cannot get those medicines for all my patients. Insurance will not cover them for all my patients. And I have to struggle giving some of my patients medications that are, decades old, whereas other patients, I can give medicines that just got FDA approved last year. And that, that's a tough pill to swallow for me as a provider who really wants everybody to get the best of everything that I have to offer. And that's the thing that I hope we can overcome by 2030, really, if you want to know the truth. And then, yes, obesity can be a symptom sometimes and not just a cause. Insulin itself, insulin therapy for people who need high doses of insulin because their insulin resistance is more profound, gain weight with insulin. As many of you know, people can put on weight with insulin. And so then obesity becomes actually worse, caused by insulin. That obesity leads to more insulin resistance, and then you need more insulin. And you can get a vicious cycle going where you give people more and more insulin and they gain more and more weight. And so sometimes obesity can indeed be a symptom of a problem as opposed to a root cause of the problem with respect to diabetes. I'll take this last one first, and this last question as the last question. How about, so I give you all some time to conclude. So the question states, I've been taking it as type 1 diabetes doesn't classically cause immune compromised situations, but they can have comorbid issues that fit that description, such as transplants and other issues that increase the risk. Absolutely, yes. That is absolutely right. Type 1 diabetes is associated with an alteration of immune function that leads to some hyperimmune responses and some deficient immune responses. And we're learning that in the context of COVID, for example, there are some aspects of dysregulation of the immune system in people with autoimmune conditions like type 1 diabetes that make them vulnerable for worse outcomes with COVID. And they're early data, but they suggest that what you're saying is right, that there is a comorbid aspect of the immune dysfunction with type 1 diabetes that can affect other processes in the body as well that relate to the immune system. And then finally, yes, I mean, of course, if people have diabetes that is ultimately addressed through transplantation, then those individuals, like anybody receiving organ transplantation, is fundamentally immunocompromised at that point to prevent rejection of the transplant. And so then they fall into a category of general immunocompromised state that has to be dealt with fundamentally at that point. So hopefully that was helpful, and I hope you all enjoyed it. And again, thanks to Danny and Alan and Tom for the invitation, and look forward to seeing you all on race day if possible. I'm having a great time. Thanks a lot.
Dr. Koliwad, thank you so much. Your expertise and time are truly appreciated. And you've inspired many of us to get back in the saddle and start raising more funds. So I appreciate that so much, and appreciate the tour of the lab as well.
My pleasure. And again, the one thing I can promise you, and I know this very well from my colleagues and from those in my lab and those who I meet around the world working on diabetes, we will safeguard and steward and use to the best of our ability every single dollar that we have the ability to spend on diabetes research, specifically and completely dedicated to stopping this disease. So you can take that to the bank if you give in any way to the ADA, that the funds that go to research, every single dollar is valued immensely by us who do research in this area, and not a single cent goes to waste. I promise you that.
Thank you. And Danny, I'm going to turn it over to you for a few words about Tour de Cure.
Sure. Thanks, Tom. And thanks, Dr. Koliwad. So as Dr. Koliwad mentioned, one of the best ways to get involved with us is through the Tour de Cure. This year, we are culminating our event on October 10th, and we are holding it virtually. Now, while an in-person event is always optimal, there is a silver lining to the virtual event in that we've removed geographic boundaries, which is great. And we've also, so you can, get friends and family members from all over the world. Last year, we had 13 countries and 27 states participate. And then also you can participate in any way you choose. And typically the Tour de Cure is a cycling event, but we have runners, walkers, cyclists. We have even a mountain climber that's joined. And then further removing the obstacles, there's no registration fee, no fundraising minimum required to join. So if you just want to do, an activity with friends and family, feel free to join us. Now it is a fundraiser, so we do encourage folks to join, and we have a minimum fundraising goal of $130 for participants. With that, you get one of our challenge accepted t-shirts, as well as if you identify yourself as someone personally affected by diabetes, at that $130 level, you'll get one of our Red Rider jerseys. At the $500 level, we have our, dare I say, collector's item, our anniversary jersey. There, you would also qualify for our finisher's medal. And then for the lifeblood of the American Diabetes Association, we have our champions. These are where 80% of our funds come in year after year, funding that research and technology that Dr. Koliwad spoke about. We have our champion celebration box. And then we're also really happy to announce that on October 8th, we will be holding an in-person champions dinner. So if you get to that $1,000 level, keep your eyes peeled for an invitation to that. And then I'd be remiss not to mention that our brand new mega champion level, which is at $5,000, well, you'll be invited to a virtual roundtable with our very charismatic CEO, Tracy Brown, and our Chief Medical Officer, Dr. Robert Gabay. Now, those of you that reach that champion level, a lot of folks like to see or understand where that money goes. So in the time that we've been doing this presentation, about an hour now, there have been 210 people that have been diagnosed with diabetes. And those people that have had that life-changing diagnosis, one of the first things that they receive is one of our pamphlets that help people begin to understand and manage it, and let them know that there is a resource out there for them. With that there's also, with that $1,000, 6 children will go to one of our educational events, or our type 1 or type 2 educational series. 6 people will get information on nutrition education, And then another 2 diabetes advocates will either go to our state or even national levels of Congress and help further the cause. So if you want to join, register, you can go to our website there, diabetes.org/Tour to Cure. Consider making a donation. It's a great way to ask friends and family members for donations. Also, starting today and for the next week, all donations will be matched by a generous donor. And those are donations of a minimum of $34 up to $2,500. So definitely take advantage of that. And then we have a goal of signing up 340 participants in the next 34 days. That's culminating on September 14th. So encourage your friends and family to join. Check out our new Tour to Cure app. It's a great way to streamline your outreach. It goes from 5 hours of outreach to about 20 minutes. And then we did have a bit of a technical issue at the very top of the hour. So I would like to actually close with the message from our Vice Chair Charles Myers. We should have the sound worked out now. And here we go.
Hi, I'm Charles Myers, President and CEO of Equinix, an avid cyclist and Vice Chair of the 2021 Tour to Cure for Northern California. Pat Gelsinger and I are collaborating to chair this year's event to create awareness around the enormous toll that diabetes is taking on our global society and help raise some money to prevent and cure a disease that impacts 1 in 10 Americans, nearly 500 million people globally. To date, the ADA has funded nearly $1 billion in research, and with your help, we can find a cure. The Tour to Cure is the premier fundraising event for the ADA, and Pat and I are asking for your help. So let's all get moving, log some miles, and raise some money for a great cause. Thanks for joining us today. If you aren't signed up for this year's event, please do so, and I look forward to seeing you out on the road.
We're very lucky to have Charles leading our ship, captaining our ship. I wanted to thank you all for coming. Tom, do you have any closing words for us? Oh, Tom, you're on mute here.
How did that happen? I just wanted to thank Dr. Koliwad again and everyone for attending. This has been one of our best sessions yet. Thank you.